Hello. Okay. Okay. So I'll make sure I'm on the right thing. Okay. So part two. Okay. Now we're going to talk about the different processes that regulate transcription. Okay. So let's start with prokaryotes. Okay. Prokaryotes in bacteria, for example, these genes are regulated into operons. Operons are groups of genes that are always transcribed together. So let me give you this example. This example right here is the LAC operon. The LAC operon is an operon that has 3 genes and they encode lactose digesting genes. Okay, so you need all these 3 genes to be turned on all at once whenever you want to digest lactose. Okay, so you might as well put them all together, right? So you organize them all together in one big chain, and basically you have a little switch and go, okay, well, I want to digest lactose and turn on the switch. Okay, so this is the LAC operon. Okay, the LAC operon is usually off. Okay, when lactose is absent, the lactose digesting enzymes are not needed, right? So if you don't have any lactose to digest, then you don't need lactose digesting enzymes. Okay, it will be a waste of energy, and more importantly, it will be a waste of protein to produce these things. Okay, so what do? Well, I don't want them expressed, so we'll build a little repressor protein, stick it right there. Okay, the repressor protein will literally bind to a region of the DNA right in front of the RNA polymerase, and it will block this thing from happening. Okay, you physically block the RNA polymerase. Okay, so it's an RNA polymerase. So the RNA polymerase is bind right there, but we can't move forward. This thing's in the way. Okay. Okay. When there is lactose present, okay, when there is lactose present, now I want to digest lactose because if I have availability of lactose, then I can digest it and I will get energy from it. Okay, so lactose, the way this repressor protein actually works, the repressor protein has a binding site for lactose. When lactose binds to it, this thing falls off the little yellow area. And when it falls off the little yellow area, then I get transcription. Okay, and I make these enzymes that digest lactose. Okay, as soon as I'm done digesting lactose, okay, the repressor protein goes back on because I have no more lactose, and I'm back to this scenario. Okay. Okay, in eukaryotes, okay, eukaryotes have a nucleus. Okay, some genes are bound up really tightly inside the nucleus, and they're called heterochromatin. Okay, they're tied up and they can't be used for transcription. Other genes are less tied up and they can be used for transcription. Okay, the most important thing are transcription factors. Okay, transcription factors. So in this picture here, these are the transcription factors. The transcription factors can turn the DNA polymerase on or off. Okay, so each cell has different transcription factors. And because each cell has different transcription factors, each cell can make different proteins. Okay. Okay. You can also regulate RNA expression, so you can change the order of introns to make different RNA. Okay, you skip that. That's not important. RNA degradation actually is. Okay, when you make RNA, as soon as it leaves the nucleus, the RNA will start breaking. Like once you make something new from the factory, okay, it will start breaking. How fast it breaks down determines how long you can keep it. Some RNAs are designed to break down very slowly. Some RNA, by slowly, it's like days and weeks. Some RNA degrades very quickly. And we're talking minutes, okay? So they can be broken down. Okay. So protein activity, they must be folded correctly. They have to reach the correct location. Okay. Okay, so a mutation can change DNA. A mutation is a change in the nucleotide sequence of a cell's DNA. Okay, so this is a point mutation. So this is the original sentence. So remember, codons are sequences of 3 nucleotides. So 3 letters, 3 nucleotides. Okay, so you can read this original sentence. You have a substitution mutation. So this is substitution. Okay, so you can sort of make up the sentence, but you can see that there's an incorrect word there. Okay, you can also frame shift. Frame shift is not substituting, but adding a nucleotide. But the problem with adding a nucleotide is now none of the words actually make any sense. Okay, there's a big chance that the enzyme or protein will still work. It might work slightly less well or slightly better, but it'll work. When you have a frame shift, you almost never works. Okay, so this is a substitution mutation. Okay. Okay. So some mutation causes these, most don't. Okay. What causes mutations? Well, most mutations are spontaneous. Okay. Every time you copy the DNA, you always copy things incorrectly. Okay. If I ask you to copy a whole book, I'll bet you there are some words in the book copied incorrectly. Okay. But there are other things that do damage DNA, and those actually do cause more mutations than usual. So chemicals, radiation, okay, X-rays. Okay, mutations are usually not harmful, and we can talk through that in class. Okay. Okay, viruses can bring new genes into cells. So the whole purpose of a virus is to infect you and get you to make virus particles. Okay. So viruses use nucleic acid, viral proteins. So what viruses do really is they hijack your cell. Okay, they take the DNA or RNA, insert it into your cell, and part of the process is to completely override your own DNA, and so it will destroy your DNA, and the cell will start obeying the virus and making viral DNA. Okay. So different viruses infect all cell types. Okay. So we'll skip lytic and lysogenic infections. Okay. But viruses can stay inside cells for almost forever, basically. So HIV. Okay. For example, has RNA genome, but it must produce a DNA copy, and that DNA copy stays inside the cell and it stays latent. So it hides inside the cell pretty much forever. Okay. Diseases. Okay. So one way to combat viruses is vaccines. Okay. So vaccines teach your immune system to recognize viruses and eliminate from the body before cells are infected, before too many cells are infected, actually. Okay. So antiviral drugs slow viral replication, okay, viroids, prions. Okay. So, all right. So hopefully, I skipped a couple of last minute things in the chapter, but they're not that important. So, okay, hopefully this makes sense to you guys. Okay.